goes from here to here. So I've gone here, here, and then I'm going to head here. Let me do this so there's no more, not extra arrows. I want to draw my own arrows for other things. Okay, so I go from step 1 to step 2 to step 3. So I've engulfed the vacuole, okay, and that vacuole fuses with a lysosome. Remember, lysosomes contain digestive enzymes, so now I'm breaking down my pathogen. So I'm breaking my pathogen down into pieces, and now those pathogens, okay, remember that's our AG here. So AG is for antigen, because you'll see that on some other pictures coming up later. Okay, so AG is antigen, AB is antibody. So the pieces of this antigen now, you see they're attaching to my MHCs and MHC1s and MHC2s. Okay, remember, all cells have MHC1s. Antigen-presenting cells have MHC2s also. Well, a phagocyte is an antigen-presenting cell. So my antigen okay, binds to my MHC1 and MHC2, and those MHC1s and MHC2s, they're moving out to the cell membrane. So now these MHC1s and MHC2s are presenting the antigen out on the surface of the cell. So this antigen-presenting cell, okay, is now going to go to the secondary lymphatic tissue. So this is no longer just a plain old macrophage. It is now an antigen-presenting cell because it's now showing off these pieces of antigens. So now this antigen-presenting cell is headed to the secondary lymphatic tissue, okay, and it's going to be looking for a matching T cell. Because remember, this is part of my cell-mediated response here. So in my cell-mediated response, I'm looking for T cells. My humoral immunity, I'm looking for B cells because they're going to make antibodies. Okay? This is cells that are going to directly attack pathogens and infected cells. So my antigen-presenting cell heads to the secondary lymphoid tissue, finds a matching T cells. When that match on the T cell is found, okay, that T cell then becomes cytotoxic or a memory cell. Okay, remember, it's got to match up both the proteins. Okay, it's got to match up both the protein as well as the MHC complex. So in this picture here, I went from 1 to 2 here. And when they match up, okay, the antigen-presenting cell is going to release cytokines. Okay, and when that antigen-presenting cell releases those cytokines, that triggers the T cell to reproduce and to make more of these cytotoxic T cells and memory cytotoxic T cells. Okay, so the binding together of the two and then the release of the cytokines from the macrophage triggers the formation, the reproduction, the cloning here of these cytotoxic cells and the memory T cells. Okay, and so this will happen when the MHC1 finds a match. Okay, so when the major histocompatibility, the antigen there that's in the MHC1 finds a match, that's going to trigger the formation of these cytotoxic T cells. Okay, and so cytotoxic T cells are going to go and attack your infected cells. Okay, so they're going to go and attack your own body cells that have been infected to prevent them from spreading that disease. Okay, so we're still working with this cell-mediated response here. Okay, so I've had my antigen-presenting cells go to the secondary lymphoid tissue, and those antigen-presenting cells in the secondary lymphoid tissue found a matching T cell for their MHC1. Okay, and so now I've started making a whole bunch of cytotoxic T cells. Well, at the same time I'm doing this, your own body cells are being infected. Okay, so your body cell is being infected by the pathogen. Okay, so the pathogen has entered your body cell, it's infected it, and same as what happened in the macrophage and in that antigen-presenting cell, the antigen binds with the MHC1. Okay, and so when it binds with the MHC1 presents it to the outside. Okay, so the MHC1 presents that infected, that antigen to the outside. So now these cytotoxic T cells that were made when I found there's some typos on this picture, just so you know. Okay, cytotoxic is not spelled right there. So these cytotoxic T cells here that were made when I activated when I got in that secondary lymphoid tissue and I found my match for my MHC1, these cytotoxic T cells that were made and released, they're going to find these infected cells. And they will match with those infected cells because they were made for that particular antigen. So when they make the match, that's going to trigger the release, okay, of perforin. Let's see if so you can see right there. Perforin, that's how you spell it there. Okay, they're going to trigger the release of perforin and granzymes. Okay, and the perforin is basically going to do that. It's going to perforate. It's going to make little holes. You can see it making little holes in the infected cell. It's going to make pores in the infected cell. And the granzymes are going to trigger apoptosis. Okay, so the perforin is going to poke holes in these infected cells. The granzymes are going to trigger apoptosis. So this cytotoxic T cell is going to kill your body cell. Okay, and so once it kills your body cell, then the goal is, to prevent that pathogen from spreading to other cells. That cytotoxic T cell is going to go find other infected cells. It is not going to just quit and give up. It's going to go find other, and that doesn't kill the T cell. So that cytotoxic T cell kills your cell and then goes and finds other infected cells to kill. Cytotoxic T cells, okay, so these cytotoxic T cells can also work on cancer cells. Okay, and so when they're working on cancer cells, it's the same concept. Okay, I've got things that are presented. The cancerous cell presents things in that MHC1 complex. Okay, and so the T cells will attack it. New proteins end up in this MHC1 complex. So these cytotoxic T cells go after that and attack it. What happens, though, is sometimes the cancerous cell ends up changing the I just made that up, what that looks like, the MHC1 complex. And so now the cytotoxic T cell can't make that dual match. Remember, it's got to match with the antigen and the proteins. Okay? And so this cytotoxic T cell now doesn't recognize that. And so that's how the cancer cells then can continue to grow and divide, is if the cancerous cell altered the MHC1 protein, and then the T cell can't find it to bind to it. Okay, so just one more illustration here of a cytotoxic T cell working. Okay, so the cytotoxic T cell here has bound to the infected cell, okay, and so it's releasing the perforin and the granzymes. So see the perforin here poking the holes in the pores into the infected cell, okay, and then the apoptosis being triggered by the granzymes. And so now the cytotoxic T cell will release and go find another one to kill. Okay, so if you notice, we've reverted back now to our macrophage. So I'm back with my macrophage that is presenting my antigens here. And in this instance, so with the cytotoxic T cells, we were binding to that MHC1 complex. Okay, and in this instance here, remember our antigen-presenting cells also have MHC2s. And so in this case, the MHC2 found a matching T cell. And when the MHC2 finds a matching T cell, that triggers the production of helper T cells. Okay, so instead of making cytotoxic T cells that are going to go out and kill your infected T cells, when the MHC2 finds a match, it's going to make the helper T cells. And helper T cells are going to go activate B cells. Okay, and so helper T cells play an important role in the humoral immunity. Okay, they're going to go and they're going to activate B cells and make those start secreting antibodies. All right, guys, I know this has been long. Okay, bear with me. We got to book it through a few more things. I'm going to start talking faster, repeating less. Okay, so now we're in the humoral stage. So we did our cell-mediated, where the T cells are directly attacking. So now we're focused on the humoral portion. And the humoral portion is with the B cells and the antibody production. So my B cells are making lots and lots of antibodies that are going to be in the bodily fluids to go find these infected cells. And so the first thing with the B cell is that it's got to bind to the antigen. And so the epitope, okay, so what's called an epitope here, that's the part of the antigen where the antibody is going to actually bind. And it can be a wide variety of things. Antigens can have more than one epitope. You can see that happening right there. Okay, they can be a wide variety of things. They can be the cell wall of the bacteria. They could be the flagella. They could be a flagellum. They could be a viral part. Okay, they could be part of the capsid. Okay, so wide variety of things. Antigens themselves, okay, so the antigen itself can be a protein or a carb. It cannot be a lipid. Okay, so it can be a protein or a carbohydrate. It will not be a lipid. Okay, and the antibodies, okay, you'll notice, remember they have the 2 receptors, okay, so they can bind to more than one antigen at a time. So they can cause a clumping happening when they're binding to these epitopes. Okay, so let's look at how we get these B cells going. So our body makes lots of different kinds of B cells, and we can go through a process that's called clonal selection to try to find the right, B cell here. Okay, and so what will happen is, remember we have those naive B cells, those ones that have not been activated, and you'll notice I've got a wide variety of B cells going on here. Wide variety of these naive B cells that have a wide variety of receptors. Okay, so the process of clonal selection is just finding the right B cell, the one that has the right kind of receptor, and then cloning that guy. Okay, so I've got a whole bunch of B cells, but it's a whole bunch of different kinds of B cells. So I need to, out of all those B cells, I need to find the right one and then start making bunches and bunches of that one. So let's step by step this then. Okay, so the first thing is I've got my pathogen, like I said, that's going to bind with my, remember, antigen is AG, our antibodies AB. So I've got my pathogen that has found a B cell that it fits with. So it's found the appropriate B cell. It's got its unique match. It's bound to this naive cell. So you notice this cell is no longer naive. It is now an activated B cell. And it's taken that pathogen in. So it's pulled that pathogen into the cytoplasm of the B cell. And when it pulls that pathogen into the cytoplasm of the B cell, it will bind that antigen there to an MHC2. So a major histocompatibility complex 2. And it will then present that on the surface. Okay, so remember, MHC2s are only found on those antigen-presenting cells, B cells, macrophages. So it's taken this antigen into the cytoplasm, different than a macrophage because it did not break and digest that pathogen down. Okay, it just took that pathogen inside so it could bind it with an MHC2 and then show it back out again. So now that pathogen gets presented, and it tries to find a match here with a helper T cell. And when it finds a match with a helper T cell, again, just like before, when I make a match with the T cell, I start secreting those cytokines. And when I make the match with the T cell and I secrete those cytokines, that makes me replicate my cells. Okay? That secretion of the cytokines when I bind with a T cell, whether it's a helper T cell, whether I'm bound to a cytotoxic T cell and I'm making more cytotoxic T cells, but that's going to make me make more of my cell. And so you'll see how these helper T cells that were made from the macrophage engulfing the pathogen, okay, those helper T cells are needed to activate the B cell. If the B cell can't find a helper T cell to bind with, it can't clone itself. Okay, and so when the B cell starts cloning itself, it's going to make plasma cells. Remember, plasma cells make lots and lots of antibodies, and antibodies have the same structure as the receptor. So they're going to go bind with the pathogen. So it's going to make lots of those activated plasma cells, and then it's going to also make some memory cells. Remember, those cells that are going to hang out, they're going to remember the antigen and be able to respond to it very quickly. So just one more picture for those of you that were struggling, that didn't like that other picture. Okay, so there's my helper T cell that got made because my antigen-presenting cell, found a match for it. Okay, here's my B cell here that has found a match that matches with that pathogen. When it binds to the helper T cell using that MHC2 complex, I release those cytokines. Those cytokines tell me to start making lots and lots of plasma cells that make tons of antibodies, and to also make a few memory cells so that next time I see this, I can fight it. Okay, so once those antibodies are made and they're out and about circulating in your cells or in your body, these are doing the humoral response. Remember, humoral is our bodily fluids here. Okay, so these antibodies are out in our humor, okay, or our bodily fluids, doing this response. And so these are some of the things that antibodies can do. They can neutralize the foreign invader. Okay, they can coat the epitope so it can't bind with other receptors. Remember, the epitope is where